Good afternoon, my good people. We go back after doing some tutorials in mathematics. We go back to biology, and today I'm happy to announce that I'm taking you through KCE 1986 biology practical paper. You might be wondering why I'm changing the examination bodies like that, but for your information, KCE was the local O-level exam that replaced the EACE after 1980 when the East African community broke. Because EACE was being done by Kenya, Uganda, and Tanzania. KCSE was introduced in 1989 to unify secondary assessment under the new 844 system. So, KCSE did not exist yet during 1986 to 1988 because the last EACE was done in 1985. So, let's look at this paper. Remember, the content normally is the same. So, whether you are doing whatever examination in biology practical, this is going to be of beneficial importance to you. So this video revisits the 1986 KCSE biology practical, a moment in Kenya's education history when science was taught with clarity, rigor, and tactile engagement. From testing wheat flour for nutrients to constructing dichotomous keys and tracing seedling surfaces, this practical embodied the spirit of discovery. In this video, we journey through the quiet brilliance of biological classification and nutritional analysis. From tracing the surface area of bean seedling to decoding the hidden sugars in wheat flour, and finally sorting marine and terrestrial invertebrates using a dichotomous key, this is a celebration of observation, logic, and life's intricate design. Before KCSE, there was KCE, and this is our tribute to its enduring impact. Let's go to the cotyledons. Question 1, you are required to have for each candidate 1 tablespoonful of wheat flour labeled specimen K5, distilled water, 4 test tubes, spatula or means of scooping, iodine solution, Benedict solution, dilute hydrochloric acid, and sodium bicarbonate powder. Also, you require to have means of heating, 2 droppers, and means of timing. Question 2 required nothing because whatever you are doing was in the question paper. And the question paper, question 2, there were the photographs of figure 1 representing animals belonging to different groups. J was a spider, K was an earthworm, L was a starfish, M was a snail, and N caterpillar, and P scorpion. Question 3, you are required to have a set of potted bean seedlings grown in light. The pot should contain at least 3 seedlings with 2 leaves each and the cotyledons intact. In this question 3, for you to have done it well, planting should be done in suitable containers, e.g. tin cans, polythene tube or bags, etc. A set can be used by several candidates. It is advisable that the teacher in charge of biology ascertain the length of time taken by the seeds to germinate and grow to the required size and shape before the examination. And here you also required 2 labels. So my guys, Here is the visual of the confidential requirements. You can see specimen K5, 1 tablespoonful of wheat flour. Then you have the photograph of animals belonging to different groups for question 2. Specimen K is for question 1. And then finally you have bean seedlings grown in light, where we have that specimen there. So now let's go to the questions, my dear friends. Question 1A, you are required to label 4 green test tubes and you name them 1, 2, 3, and 4. In each of them, you place a small pinch of the specimen K5 into each of the test tubes. Carry out the following food tests, record your observations and the deductions in the table below. Test tube 1, you are supposed to add 3 to 5 drops of iodine solution and observe. Test tube 2, you add 2 mL of Benedict solution and shake. Heat gently and observe. Test tube 3, you add 2 mL of dilute hydrochloric acid. Shake and heat carefully to boil. Remove the heat and add sodium bicarbonate powder in small amounts at a time until there is no more fizzing. Add 2 mL of Benedict solution. Heat gently and observe. Test tube 4, add 2 mL of water and shake to mix with specimen K5. Then add 2 mL of dilute sodium hydroxide and shake to mix well. Finally, add 2 mL of 1% copper sulfate and mix well by shaking. Let the mixture stand for at least 5 minutes and then observe. So we have our good table here, where you have the test tube number, the observation, and the deductions. Test tubes are number 1, 2, 3, and 4. In part B of question 1, you answer the following questions. What is the role of the following in test tube 3? Hydrochloric acid, Roman 1, and Roman 2, sodium bicarbonate powder. Fine. As usual, I told you I will be dealing with the question. I changed the format. I do the question, then I give the answer immediately. Before, the format was I do the 3 questions and give the answers. But now I do the question and give the answer. So question 1 answers. Recording the observations and deductions in the table below. Test tube number 1. The observations were dark blue, or you could have seen black, and the deduction is starch present. Number 2, slight change from blue to green, and you deduct that there is presence of small amount or traces of reducing sugar, or you just say no significant amount of reducing sugar. Number 3, the observations were slight change from blue to green to yellow or orange, and the deductions were non-reducing sugar present. Or, you say, there is a great amount of reducing sugar present. Or, you say complex sugar has been converted to reducing sugar as your deduction. And finally, number 4, the observations you had were pink color, purple in some cases, or violet, or you say mauve color forms with the time, any of those colors. So here, you deduct that proteins, or you can say amino acids, or you can say peptides or peptones present. And finally, B, the role of the following in test tube 3, hydrochloric acid, Roman 1, it hydrolyzes disaccharides, therefore making it possible to test for reducing sugars thus produced. And then, for sodium bicarbonate powder, it neutralizes the hydrochloric acid or makes the solution neutral. Benedict's test has to be done in a neutral solution. So, in Roman 1, you can see that you are having 2 marks. When you say hydrolyzes disaccharides, you are given a tick. And when you say, therefore, making it possible to test for reducing sugars thus produced, you get a tick. For Roman 2, you had only 1 tick. That is, it neutralizes the hydrochloric acid, or you take the other alternatives. We are through with equation number 1. We come to question number 2. And the question was, the photographs in figure 1 below represent animals belonging to different groups K, L, M, N, and P. Using observable features only, construct a dichotomous key to sort out animals into the 6 different groups. The dichotomous key should start with the first step, 1A, 1B, given below. Continue to construct the rest of the key as necessary to sort out the animals into separate groups. So there you have the figure 1, where you have the J, K, L, M, N, and P shown very clearly. So it is up to you to make very keen observations so that you put all those animals into their different groups, or you identify those animals using the dichotomous key. So here they have started for you the dichotomous key, where they start with 1A, body segmented, go to 2, and B, body not segmented, you go to 3. So from there now, continue with the story until you classify all the animals. B of question 2. Assume you have just discovered an animal Q shown below. The animal is shown there below. Into which of the groups which you have sorted out in A above would you place such a newly discovered animal? Give reasons for your answer. I'm telling you, they tested the scientific knowledge. Let's now go to the answers of question 2. Continue, where A, you are continuing to construct the rest of the key as necessary. So what is in red is what was given. That is 1A, body segmented, you go to 2. B, body not segmented, you go to 3. So continuing that key, number 2A, legs present on each segment, you have identified N. Number 2B, legs not present on each segment, you have your K. You have identified. Then you go to B, 1B there up, you go to 3, and when you go to 3A, you have, with a shell, you have identified M. And B, without a shell, you go to 4. 4A, star-shaped, L is identified. B, not star-shaped, you go to 5. And 5A, with sting or pincers, P is identified. Without a sting or pincers, J is identified. And you end up your dichotomous key. Remember, dichotomous key is where you identify animals looking at their contrasting characteristics. I also discovered that you could have used a slight difference to that key original given by the examination body. And we start just the way we did. You are given 1A and 1B, body segmented, you go to 2, and body not segmented, you go to 3. So continuing the key, you have 2A legs or prolegs present on the segment. You identify N, so you have added prolegs there. B, no legs or prolegs on segment, you have K identified. So we go back to 1B and go to 3, 3A with a shell, as we had M identified. Without a shell, you go to 4. 4A, star-shaped, L is discovered. B, not star-shaped, you go to 5. So in 5 is where we brought a few changes, and we say 5A with the crow-like appendage or pincers. So the crow-like appendage is an added advantage to your biology knowledge. And B, without crow-like appendage, you get J. So you identify P and J with that characteristic. B, groups sorted out in A above to place the newly discovered animal. The groups are you either place it in J or you say P and J as your answer. Reasons for the answer is that it has 4 pairs of legs arising from the thorax, or you say cephalothorax. Also, abdomen is not segmented. All right. Legs is segmented or jointed legs. Body divided into 2, cephalothorax and abdomen. Body segmented, except any 2 reasons. Ladies and gentlemen, we now come to question 3. It is getting dark here. Examine the bead seedling provided and answer the following questions. A, draw a diagram of one of the seedlings provided and label its different parts and state the magnification. You get 2 marks for the magnification. The pair of first foliage leaves and the cotyledons are used for certain nutritional functions. One contribution to these functions is dependent on the surface area of each structure. B, Roman 1. From one of the seedlings, detach the first pair of foliage leaves and label them as specimen D. Remember, you had been given labels. Also, detach the cotyledons and label them as specimen E. Bearing in mind that one small square on the graph paper provided has an area of 1 millimeter squared, use the graph paper to determine the total surface area of the leaves and then of the cotyledons. So you give total surface area of leaves and the total surface area of cotyledons. So, ladies and gentlemen, the graph paper to determine the total surface area is displayed there, as you can see. Roman 2. Explain briefly the method which you use to determine the surface areas. You get 5 marks. Roman 3. From Roman 1 above, calculate the ratio of surface area of leaves to that of the 2 cotyledons. Leaves is 2 cotyledons, you give the ratio. B. Explain the significance of the ratio obtained in B3, Roman 3 above, in relation to the nutritional contribution of each structure to the plant. 6 marks. And C. Predict what would happen if such a seedling growing in garden at this stage of development and the leaves and the cotyledons practice from it. And if the seedling growing and the apical bud removed. So now we are in for the answers, guys. Question 3. Label the diagram of one of the seedlings there. You can see the diagram there. We have labeled first foliage, leaves, epicotyl, cotyledon, or you can call them cotyledonary leaves. We have hypocotyl, or you can call it a stem there, but we have the hypocotyl below there. Double lines must be shown for the mark to be awarded for the stem. Then we have the main or primary or taproot, which should have a double lines must be shown. Then we have the secondary or lateral root. Drawing 2 marks. Correct labels half each. And you have 5 of them, so 5 divided by 2 is 2.5. You round off, you get 3. Magnification lies between times 0.25 to 1.5. And here, magnification, you are guided that it is size of the drawing over actual size of seedling. And you use a ruler to measure both the drawing and actual seedling, and you express them as a ratio. All right. Now, B, to determine the total surface area of the leaves and then of the cotyledons. 1, total surface area of the leaves. You are going to get within the range indicated in the supervisor's report. Examiners were to calculate the largest and the smallest leaf surface. So that information was with the supervisor's report. Total surface area of the cotyledon was within the range indicated in the supervisor's report. Examiners to calculate the largest and smallest leaf area. Roman 2, examining briefly the method which was used to determine the surface area, 2 marks. The first one is trace or draw the leaf outline on the graph paper, tick. Count the number of squares, tick. Round up half or more squares to whole number. Multiply by 2 to get surface area of a single leaf. Multiply again by 2 to get the surface area of the 2 leaves or cotyledons. Or multiply by 4 since 2 leaves have 4 surfaces. Roman 3. The raw data should be calculated, ratio calculated, and expressed as leaf is to cotyledon. I think you would like the way I put what I've given as the marking points of the council clearly for your own revision purposes. So this part tests your ability to estimate surface area by tracing outlines on graph paper. And therefore now, steps to follow. So, guy, detach the first 3 foliage leaves. Label them as specimen D. Detach the cotyledons. Label as specimen E. Place each specimen flat on graph paper. Trace the outline of each leaf and cotyledon carefully. Count the full squares inside the outline. Estimate partial squares, e.g., 2 half squares is equal to 1 full square. Add up the total number of squares. Each square is 1 millimeter squared, so your final count gives the total surface area in millimeters squared. Example format for the answer: total surface area of leaves of specimen D, e.g. 320 millimeters squared, or total surface area of cotyledon specimen E, e.g. 250 millimeters squared. C. Explaining the significance of the ratio obtained in B3 above in relation to the nutrition contribution of each structure to the plant. The foliage leaves have a large surface area to allow more photosynthesis, or once to that effect. The foliage leaves obtain more sunlight, or you can say the foliage leaves obtain more carbon dioxide for photosynthesis. Note, the smallest surface area of the cotyledons provide stored food. The stored food is significant in early growth, or it is significant in germination. Later, the cotyledons help with photosynthesis. When they turn green, chlorophyll is formed in them, and you have observed that during planting after they germinate. D. Predicting what would happen if such a seedling growing in a garden at this stage of development happened. Roman 1. The leaves and the cotyledons plucked from it die or reduce growth rate. Please reject wither alone and dry alone. Don't say when you pluck they dry. All right? Roman 2, when you remove the apical bud was removed, growth will cease. Although the plants may stay alive for some time. And the total marks is 52, maximum 50 marks for this examination paper. What is the educational purpose of question 3? The question reinforces functional morphology. By the way, guys, morphology is external appearance. Cotyledons store nutrients. Foliage leaves begin photosynthesis. Surface area relevance. Large surface area enhances light absorption and nutrient exchange. Practical skills: tracing, measuring, and interpreting biological structures. So guys, thank you for listening. And I conclude by saying this. Before KCSE shaped a new generation, KCE laid the foundation. This practical reminds us that science is not just about answers. It's about asking the right questions, observing with care, and honoring the systems that brought us here. May this video rekindle pride in Kenya's education journey and inspire future learners to see biology as both logic and legacy. I won't forget to acknowledge the students and the teachers of 1986. Your hands held seedlings, your eyes traced specimens, and your minds built the frameworks we still use today. This video is dedicated to your legacy and to every learner who continues the journey of discovery. Credit goes to content by Mr. Thomas